Hello, I am Colin Schuman and I'm in Bio 200 Lab. This is a presentation on the gut microbiome. Microbiome can be defined as the location where different types and proportions of microorganisms live and operate. More specifically, the gut microbiome is the different types and amounts of bacteria present in the gut of an organism. The gut microbiome can reveal a surprising amount of data about an organism, and some studies have even found a link between the microbiome and allergies in organisms when sequencing and recording the bacteria present. Microbiomes have even aided in diagnosing and studying the cases of autism in subjects. Certain studies have in fact found a link between how gut microbiome interacts with the central nervous system and the symptoms of autism. Some studies have shown that there's a difference in the gut microbiome composition for individuals with autism versus those who do not. There's a correlation between a species in a particular phylum with autism and then individuals without autism who would have a smaller proportion or larger proportion of that species. These studies are correlative rather than causal, where the bacterial species do have a direct correlation with whether or not an individual has autism. In this project, we looked at the fecal sample of two types of reptiles, a corn snake and an eastern box turtle. We predicted that the gut microbiome of the snake would have a higher amount of protein digesting enzymes than the turtle due to the fact that it has a protein-rich diet compared to the turtle. The female corn snake, Autumn, was 10 years old and dieted on mice and small rats. The 7-year-old female box turtle dieted mainly on vegetables. Both of these reptiles were in very good health and lived an indoor lifestyle. To find the composition of the gut microbiome, there's 3 major steps. First, you have to isolate the DNA from the fecal sample using a commercially available kit, and then after that, you have polymerase chain reaction, or PCR reaction. Those are done to amplify that particular region that we isolated. After that, we amplified regions of the DNA, which is going to be quantified and tagged with a particular index tag. After that, the third final step is going to be determining the nucleotide sequence of those amplified regions using a next-gen sequencer. We get more particular in purifying and concentrating DNA by isolating the DNA from fecal matter. We used a commercially available kit from Qiagen Incorporated called the Qiagen kit. We used that to purify DNA, where you use a stool sample and a buffer, which prevents enzymes from creating the DNA, and the DNA binds to a filter, goes through a column, and it washes away the impurities, in turn allowing you to isolate the DNA. That purified DNA is then concentrated 10-fold using another kit called the Zymo kit from Zymo Incorporated. This concentrates the DNA 10 times. We then use the absorption spectrophotometry to determine the concentration of our DNA, and we wanted to see if there was any contaminating protein or RNA in the sample. This was determined using the nanodrop spectrophotometer, and we did not just check the concentration, we also checked the absorbance values. We checked it at 260 nanometers, and then we compared that to 260 to 280 to see that ratio. We checked that concentration when we're given the volume. We had to determine the proper amount to put into 100 milligrams of that sample so that we could do proper dilutions. This allowed us to determine if the dilution was required. Once we determined that the concentration of the DNA group performed was adequate, we performed amplicon PCR to amplify the regions of the 16S ribosomal RNA gene. Specifically, to amplify the V3 and V4 regions, because that's the particular part in the RNA gene that's particular to the bacterial samples. So we can check those regions and check into a database to see what the actual composition of the DNA was in our sample. After we amplified that, we moved on to gel electrophoresis, where DNA fragments of different sizes are separated using an agarose gel. This assured that we amplified our DNA properly. In particular, our gel set was at 250 volts because that's what the kit and procedures said. All the procedures were done in accordance to the manufacturer's instructions and in accordance to our lab manual. We then moved on to our PCR cleanup reactions, which clean up and remove the components of our amplification buffer, which contain lots of different chemicals that impeded with the actual DNA component. We needed to use them as a buffer for the amplification PCR as well as forward and reverse primers. These were removed along with cleanup reactions. The indexing PCR added a unique set of short sequence tags to the ends of amplicons for each of the samples so that we can differentiate the samples. After the indexing reaction, we cleaned it up again just to remove the same components as the last reaction, like the buffer and the foreign reverse primers. Afterwards, we used a fluorometric assay to determine the concentration of our amplified sample. We used the fluorometric assay because it's much more accurate than the spectrophotometer. Finally, we used the next-gen sequencer to determine our nucleotide sequence with a DNA analyzer. After we finished our reactions, we looked at our results from the nanodrops. We used gel electrophoresis, which is right here, shown on the graph, and we have our base pair length goes from 100 at the top to 4,000 at the bottom. And none of our readings showed between 500 and 800 base pair lengths, which is what we wanted. This indicated that we did not have DNA present, so something went wrong. The concentration for our PCR calculations helped us to determine the volumes for the regions of the PCR reactions, like the master mix DNA, control DNA, and the DNA itself, with just water. We also had a positive and negative control on this. Our positive control was pure E. coli DNA, and the negative control was just water. We then moved on to PCR reactions, which was clear that we did not effectively amplify our regions. None of our base pairs fell within 500 to 800 range. For that matter, we didn't even get a reading, not even for a positive control, which shows that something went wrong in our amplification process. We then moved on to indexing PCR and cleanup. From the fluorometric results, we found that each of our samples had a concentration above the required threshold that we needed for our samples. So basically, you have to have a certain reading, and if that concentration wasn't good enough, we could not continue with the data. Unfortunately, our data was not showing any DNA presence. However, we didn't actually have our results to use the other animals, so we actually had to compare the composition of animals collected from previous years. Our class was unable to use our own samples that we conducted due to the fact that they were not done getting sequenced. We had to use samples from previous data, so instead we used results from a 3-year-old female short-haired guinea pig, ID 322, and a 3-month-old female husky, ID 343. We had to rewrite a testable hypothesis for our new data samples. We did classifications at the kingdom level, order level, and genus level, which is a distribution of the composition of bacteria species at different levels. At the kingdom level, we found that overwhelmingly majority of the samples fell within the bacterial kingdom. At the order level, we found that both animals had started to show their classification differences. There was not much correlation between the two order levels within the husky and the guinea pig. At the genus level, we saw an increase in unclassified species. However, our analysis of the sequences showed that the husky has higher concentration of bacteria such as flavobacteria. This is not surprising, as the husky is an outdoor dog that would easily come in contact with flavobacteria, which are very common in soil and fresh water. Another factor to consider is the high amounts of enterobacterials found in the gut microbiome of the husky. Enterobacterials have been linked to gastrointestinal issues, and this corresponds to the data given on the husky, since it was recorded to have a case of coccidea in the past. The bacterials were found exclusively in the gut of the guinea pig, which is puzzling due to the fact that bacterials are usually found on skin and in the gut of all animals, not just guinea pigs. That's it. Thank you.